here, then they don't run anymore. They control everything remotely. But the nice thing is that they were able to see what the mouse actually sees. And this is the perspective of the mouse, right? The mouse normally runs, so it's like there is no really a set direction, but it's like a more two-dimensional thing. And then if you, so what in this paper and the works people did was to look at the spectral distribution, spatial spectral distribution of information in this scene. And, but we don't need to do like the specific quantification to try to guess what we are going to get, right? So we know that from the sky, we expect a lot of ultraviolet light, so we expect a lot of UV. And in the ground, we expect to see like more green spectral information. Okay, so they did the analysis. And they also look at the retina of this animal. And especially, they look at the distribution of photoreceptors on the retina of the animal. So these guys encode information upside down. So this is the ventral side. So this is the part that is looking to the sky. And this is the part that is looking to the ground. So you can see they only have two cones, right? And the one that encodes for UV is actually highly dense concentrated in the area that encodes for the regions where you find more UV light, right? And the other way around. So this is really cool. And this is, again, some efficient coding theory people trying to make predictions on biology, what we observe, the things we are observing, and kind of try understanding and like bridging a gap between function and why we have this function. So there are also some works in the case of zebrafish. So they also have found some spatial distributions of photoreceptors. Here the case is less clear because the animal also can navigate like in the set direction, but still they were able to find like some regions of concentration of photoreceptors. Then this answers a question about how the, what's the optimal or more efficient distribution of opsins that you can have on the retina. But this is not telling us anything about the, how these opsins encode the spectral information. So this is a question that we had. And then this question was inspired by some experiments. So again, experiments became fancier. So people now are able to manipulate genetically animals. So they put some calcium indicator in the animal, and then you can check in the living animal, checking what neurons are active only by using your microscope and just like checking, looking at of course you need to do genetic manipulations before, right? But you can see what neurons are doing in the brain of the animal. So this is what these people did here. So this is the sensitivity of cones as a function of the wavelength in the isolate photoreceptor. So they take the neurons, then they measure the sensitivity, and they get these curves. But then what people did in vivo was to check at the responses of these photoreceptors in the network. So this is the difference. So here are only like the separate photoreceptors, and here the photoreceptors are within the network. So there is the interaction of these guys that you see in white that are called the horizontal cells. And what they found is that if you kill the talk between horizontal cells and cones, then you start having something like this. So to get these functional curves, you really need the influence of horizontal cells. And something important about color vision is that if you only have, so if, for instance, you have one of these cones, right, you can't really tell apart the difference between light intensity and light frequency, right? So for instance, if you have this cone here, you can have like some specific response at this specific wavelength. But assume, for instance, if now you change the wavelength and you also increase the intensity of the light, right, your neuron might be able to give you exactly the same response. And without going to changes in luminance, if you send a stimulus here or a stimulus here with different wavelengths, the neuron is going to give you the same answer. So you can't really tell apart frequencies from a single neuron. You need to have interacting neurons. And the solution, this is like color theory, and what we humans do is to do something that is called color opponency. So we compare the different the signals from different cones, right? And we are able to discriminate specific frequencies from there. And this is a little bit how this looks like, right? So you can see, for instance, in this red response, so you have some positive response of the neuron and some negative response of the neuron, right? Then it already tells you that if the neuron depolarizes or hyperpolarizes, then you have one or the other wavelength. You don't have these compounding responses for different ranges of the wavelength. And even then, more interesting, going to the naturalistic inputs of the animal. So what these people did was to put some cameras in on the place where these animals live, a hyperspectral camera, and they look at the spectral information from this environment. So this is a camera in space. So what they did was to use a principal component analysis. So you just have a high dimensional data, and then you try to collapse the data into the axes or the axes that better explain the variance that you have, right? So if you don't have a low dimensional data, then you can't. But in this case, they found that with the three first principal components, they can explain most of the variance from these typical environments. And the interesting thing is if you qualitatively compare the responses of your neurons with the principal components, they look similar, right? So one of the questions that we had was whether the functional responses of these neurons and networks in the zebrafish were actually highly tuned by this type of information in the environment. So what we did was to propose a dynamical model. So we have homogeneous populations. So for instance, this is a case of a dichromatic population. We only have red and green cones. And then because we have this experimental data showing that horizontal cells are quite important to drive this behavior, then we also include a horizontal cell type. This is an inhibitory population. And then we allow this network to interact with some specific weights, right? We can do this for dichromatic, trichromatic, tetrachromatic systems. So this is a framework that allows us to investigate not only the case of zebrafish, but also what's going on with other type of animals. And the first question that we had was about the specific structure of the network and the connectivity of this motif. So the first question was experiments show that the horizontal cells are important, right? But cones can also talk to each other. So we could have some excitatory inputs going on in the network, and we wanted to know what were the effects and the benefits that you can have if you include these excitatory inputs in the network. So what we found, I'm going to show you only the results for the dichromatic case. I'm happy to talk about this for the other type of networks at the end. But you have to the only thing I have to say is that this generalizes to bigger networks. So for the case of the dichromatic, what we did was to put then different combinations of opsins, so red, green, blue, green, right? And then we put the response curves into our dynamical systems, and then we checked for fixed points. So we started to then send a specific color inputs to the network to see if these networks were able to discriminate specific colors, or if you send two different spectral colors or spectral stimuli, then you can also have this compounding thing that I explained in the beginning. So what we saw is that if you have a weak or zero excitatory inputs, right? So we allow the network to change the weights. So you can have excitatory and inhibitory inputs. And then we found that in this regime, then we get always a unique fixed point. It means that if you send a specific color, the network is able to tell you, OK, this is red or green or blue. But as soon as you increase the strength of the excitatory inputs, then you start to have a bistable thing. So this will tell you that assume that you have a combination, you are sending a stimulus, and then you have a combination of different colors. Then depending on what was the previous color that you have, then you might find that you discriminate, instead of saying red, you might say green. So it doesn't really give you a really reliable system to discriminate colors. So what we really want to do is to have this thing. So with this approach, then we were able to understand why we actually have these horizontal cells playing a role in the zebrafish. Then, so this is just the conclusion that I just said. And then the next questions that we wanted to do, so all this previous thing that I showed was for non-naturalistic input. So we were just using poles of monochromatic colors and trying to see if they can, if neural networks can discriminate this. But now we were interested in understanding the efficiency of this type of networks at encoding the typical environmental information that the zebrafish encounters, because we have the data, right? So this we did in two main branches. So the first one was to try to say, okay, so let's play with networks. Let's try to include more neuronal types and try to change the weights between these neuronal types and see what's going on. So I'm not going to tell you the whole thing, but I'm going to tell you the final result that we got. So these excitatory cone populations can generate the three principal components that you normally find in the zebrafish environment, with the influence of two inhibitory populations, two horizontal cell populations. So something that the experimentalists found is that you don't have only one type of horizontal cells, but you have several types of horizontal cells playing a role in the color opponency of these guys. So we were able then to explain the data by saying that these networks actually are able and highly efficient at encode this type of principal components and not only monochromatic inputs. And then the second question that we wanted to do is to make is to understand the evolution of the opsins itself. Because everything I have shown to you is based on the fact that the zebrafish has 4 opsins, and they have this specific tuning to these frequencies, right? So what we wanted to do was to change the tuning of these different opsins and see how efficient will be these fake networks that to encode this very same information from the environment, right? So we did that, and what we found is that the more efficient networks improve a little bit the information encoding of the zebrafish, but not much. So it was like a 10% increase in the error coding of the two cases. So in general, zebrafish is actually doing a really good job at fitting the principal components that they find in the environment. So once more, we are able to understand a little bit from the efficient coding perspective why we observe the things that experimentalists are measuring in their data. So then I hope, up to now, I convinced you that animals have a specific trade-off, right? So they really want to, well, first here, they really want to encode important information from the environment, right? Because they need to survive, so you have something they really need to do this encoding of information as much as you can. But they are also constrained by the specific substrate that they have. So, for instance, you can have, in the previous example of color, you can tell me, okay, so, but you only had 3 cones in your 3 neurons in your system, right? But you could also have many neurons narrowly tuned, right, to that specific frequency. So you never have encoding of different frequencies because you don't have this broad distribution, right? So you could have many, many, many neurons. But then what happens is that this is not possible because neurons are really expensive, right? And then you need to get information from these many neurons, and then this is going to be energetically cost. But if you have a better solution, like only having 3, and making sense of the information that they are sending, that it is much better, right? So there is definitely a trade-off between energetic cost and the environment and the information that the brain wants to encode and process. So then I want to tell you some more recent work we are doing. And this is, so I told you that this is like the sketch of the retina. So I told you that neurons encode information in this hierarchical way. So you have photoreceptors. Then if you are in the mammal retina, then you have like the bipolar cells. Then you have the ganglion cells, right? And then it was like kind of homogeneous thing, right? But then I didn't tell you anything about what happens within the layers. If you actually look, for instance, these are the names of the neuropiles in the fly, but this could be like bipolar and ganglion cells. Then you see that there is going to be a diversity of neuronal types within the single layer of neurons, right? And the thing is, so here is the example. So if you have the mouse, then if you look at bipolar cells, again, experiments are fancy. So what people are doing now is to like manipulate the mouse and then put some natural or a plethora of non-naturalistic stimuli in front of the mouse and check what the neurons are doing. So here they have like a lot of neurons, right? Bipolar cells active. And what they do is look to try to make sense of the dynamics of these neurons. So they do a clustering algorithm to understand and to classify neurons by function. So here you can see that there is a large amount of bipolar cells, and there is an even large amount of ganglion cells that they are able to identify from this functional analysis. And if you go to the case of the fly, then again you see like within the lamina, you have differences of temporal filters or processing functional properties of neurons. And if you go downstream, then you start having more and more of this diversity of neurons. Then I told you before that neurons are costly, right? So you don't really want to make like a huge amount of neurons because you can't keep that energetically. So one of the questions in the field is why do we actually have that specific number of neurons? So why do you have this specific number of bipolar cells or ganglion cells in the retina? So what we want to do is to try to understand this diversity of neuronal types from the coding perspective, right? And to do that, then I want you to imagine that you are this fly, and then you are navigating in this environment, and then you are looking at the side. So you start having like some moving leaves, right? And then suddenly you do a body saccade, and you start looking at this thing. So you see that the dynamics and the type of inputs that you are getting from just like switching a little bit your position are super different, right? So how can these animals actually make sense of these highly complex and different naturalistic and dynamic inputs is one of the questions. So if you actually look at the distribution of, for instance, contrast or luminance of this type of motion, naturalistic motion, you find something like this. So you find that the probability for instance, so if you look at the video, you can look at a pixel, and then you can follow what's going on to the pixel, and then you can extract the luminance or the contrast and make a probability, and like put the probability distribution or the distribution of these quantities, right? So what people find is that these functions are not non-Gaussians, right? So non-Gaussian, so they really have important correlations, right? And if you plot this in the Fourier space, then you compute the power spectrum, you see that they follow a power law. And then this power law goes normally from an exponent of -1 to -2, right? And this is what people have found from the natural scenes perspective. So this is then what we want to try to characterize in our framework. So we want to say, okay, so if we send this type of correlated inputs into our network, right? What are the typical temporal properties that we are going to find in our neurons? And are these same temporal properties that we find in our theoretical framework the things that we are finding in the real animals? So to tackle this question, okay, so this is like the hypothesis. We hypothesize that this diversity makes sense if you have naturalistic inputs. Then we want to test, you can, maybe you are asking, like, why is she talking to me about the flies? So flies are boring, are annoying, and they are not really interested. But I can tell you that they are interesting, and like we are entering this like new field of connectomics. So some of you already talked this week about this connectomics area. And the fly is a really, really, really interesting organism. So it's annoying, and it's small. And because of that size, people have been able, so people, the Flywire consortium by Princeton, and also some people in Geneva, they were able to take the brain of the fly, to slice the brain, and then to record the specific neurons that, of that specific brain. And then they took all the data. Initially, it was humans actually looking at the images and saying, this connects to this connects to that. So it was like a huge human effort, right? But then, of course, like machine learning and like got into the place, and then they started to say, okay, maybe we can just put this into a machine learning thing, and then the algorithm will tell us what are the connections between the neurons, right? So they did that, and now this is a huge and super cool data set in which you can go to a specific region of the fly, of the brain of the fly, and then, for instance, if you are interested in some specific neuron, you can go check for that neuron and see, for instance, what are the connectivity patterns of this specific neuron. So this is a super cool data set, right? And this was done for one single brain. And now I think they already released the other brain. So now we have like 2 brains to also compare intervariability across species. And then this is, I think these are like different neurons, like neuronal, just like to distinguish further. This is the full brain of the fly. Yeah. It's all the brain. So whatever you want to investigate, like olfactory or mushroom body, you can look at the neurons in this. So then I'm presenting this because I'm trying to tell you that flies are interesting, and that's why we are using flies. We are trying to also make sense of these ideas from the connectomic perspective. And the other reason why flies are cool is because we have a lot of data. So this is the optic lobe of the fly. I already showed you the mammal retina, but this is what the fly looks like. So we have photoreceptors, we have some lamina neurons, medulla neurons, and the lobula plate neurons. Again, they do like this hierarchical processing of information. And then what we can do, and we have done, is to look at different data from the different research papers, and we have tried to characterize what are the typical temporal responses of the neurons. So we look at neurons from this linear, nonlinear model that I showed you before. So we are going to characterize them by some specific temporal filter and look at what's going to be the output of the neuron based on the input signal that we send. So we then were able to find like 2 main types of filters. One that we called bipolar filters, meaning that they have like a positive polarity, and then they send like they have a negative polarity, and other ones that have like a single polarity. So this is so we characterize the neurons with 2 main features. One is the area of the filter, and the other one is the half-life time scale, meaning the time at which the neuron integrated half of the area. So this is going to be like the characteristic time of the neuron. And then I'm going to here I'm plotting like the different data points of the half-life time scale as a function of the filter area. And the different colors, for instance, red and yellow, are differences between the data and the fitting function that we are using. So we put, like, we want to describe this mathematically, right? So we use a mathematical function and we fit the data. And then we are able to describe well the neurons in the fly. And then we want to go to our efficient coding. So this is just like an illustration for you to like have an idea of what this looks like. So we have the input. So as I said, we have some input with correlated noise. So this is going to be like what people have found in natural images. Then we are going to find, like we are going to have like the model of our neurons, linear, nonlinear model, some temporal filter, some noise. And this is going to give us the response of each neuron, right? And then we are going to have a decoder that is going to tell how important are going to be the responses of each of these neurons. We are going to optimize this all together, right? And then this is going to give us a final output of our system. And then what we want to do, this is efficient coding, is to compare the output of our network with the input of our network. And if the amount of information, we can compute mutual information, minimal square error. So if this error is small, then it means that the network is doing a really good job at encoding the information. So then what we did first was to look only at the neurons in a single layer of the fly. So we took the lamina neurons. These are the typical filters that we find in the literature and that we can fit, right? And we put them into the networks that I showed you before, right? These are like, we can play with combinations and with network sizes. And what we do is to compute the signal-to-noise ratio. So this is like inversely proportional to the mean square error, and this tells you how much of the signal your network is actually able to decode. And then you can, this is for combinations of triplets of neurons, right? So we are working with 3 lamina neurons, and this is what we get. So we get that the signal-to-noise ratio is small if you have highly redundant units. But as soon as you start to have the distribution that you see, like in the fly, these highly diverse neurons, then you start to having a better performance in signal-to-noise ratio. Then we start to play with more than 3, with different network sizes, and also we define the diversity coefficient to be able to compare this signal-to-noise ratio across different networks. So what we do and what we find is that in general, so you have that as soon as you start increasing the diversity coefficient in your network, so you increase the number of neurons, but also the diversity of temporal properties in the network, then you start having a high signal-to-noise ratio. So this is a trending that we observe in the network. Okay, so this somehow explains why, for instance, fly. And an important thing is also that you find a saturating behavior of the signal-to-noise ratio. So this tells you that maybe you can include more neurons to gain a little bit more of signal-to-noise ratio, but maybe with only 3 neurons, then the fly is actually doing a good job at encoding this information. So the next thing then that we wanted to do was to try to generalize these ideas beyond the typical filters that we find in the fly. So here, what we did, we have the mathematical function to fit the filters of the fly. So what we did was to change parameters to fill all these blue and red regions to describe different type of potential biological filters in the fly. So here you see some examples of some filters that are not in the fly, but kind of look like the filters that the fly has. And then what we wanted to do was to compare and to look at the coding of these neurons in this network format. So what we did was to look now at pairs of neurons. So now our network is a pair of 2 neurons. And then we are going to look at the signal-to-noise ratio. And because we are interested in the diversity of these networks, then we are going to compare these 3 scenarios. The first one, in which we have 2 monopolar-like neurons, so these ones that are nearby in diversity coefficient. Then we are going to compare 2 bipolar neurons, so these ones that are within the blue region. And then we are going to compare like across these 2 clusters, right? This will be like the really high diverse networks. And then what we want to see is for not only for the few data points that we have in the fly, but for all the data points that we can generate with the fitting function, then if our results generalize for this case. And then what we find, okay, this is, what we find is that yes, so if you have a highly diverse network, then you will improve the encoding. So this is a plot that shows again, like this is the feature space, half-life time scale as a function of the filter area, right? And what I'm doing here is like, I'm going to take a neuron that has this specific area and this specific time scale, right? And I'm going to put this neuron in a network with all other possible neurons that I have in this feature space. So I have different neurons, right? So I can compare this scenario, right, in which I have this neuron here combined with other neurons in this thing, and I can compute the signal-to-noise ratio. And the signal-to-noise ratio is the color that you see here. So the higher the signal-to-noise ratio, the better. So what this plot is telling you is that for a general amount of filters that you can generate in your network, if you have a high diverse network and that combines this bipolar and these monopolar neurons, then you increase the signal, then you maximize the signal-to-noise ratio, which is in this red region here, right? And the even nicer things that we show is that if you overlap the data from the fly, then we get that this was a biological neuron in the fly, and it tells that if it combines with one of these biological neurons here, it maximizes the signal-to-noise ratio. So this seems to explain a little bit why we see these specific phenotypes in the temporal properties of the neurons, right? And it also tells us that actually if you have this diversity, high diversity of temporal properties, then you can encode better the input signal that you have. I'm not showing this, I'm not going to show this here, but we have also done this for different stimuli types. So we have, for instance, decreased the correlation of noise and try to go like into the Gaussian regime, so you don't have really naturalistic inputs, right? And what we find is that this advantage of the combination and diversity of networks is actually completely lost. So you can do as well as the combined if you have two monopolar neurons or two bipolar neurons. So this actually seems to be something that is driven by the complexity of natural inputs that this animal normally encode for. So we are also now trying to make sense of all this data using the connectome. So we are testing our efficient coding hypothesis. I'm not going to talk about this now, but I can talk about that later. And then the idea is to use this rich data set to understand, so first to test our hypothesis on this efficient coding, and also for us to be able to explain some of the patterns of connectivity that we observe in these flies. So with that, then, I just want to conclude the talk, and with this quote by Barlow that tells that a wing would be a most mystifying structure if one did not know that birds flew. So the idea of the efficient coding is like to tell us what. So we are interested in understanding a specific phenotype. But to really understand and for us to make more sense, we need to put this phenotype into our context. And this is what the efficient coding is doing, and what the type of questions that I'm trying to understand in the sensory systems. And yeah, with that, I just want to thank you and finish.
For this nice talk, and we start here.
Oh, thank you for the talk. I was thinking during your talk, I mean, efficiency is obviously important, but there is a thing about biological systems is that usually if something not goes well, you cannot replace it. So they also need to be resilient. And these two things usually are in a trade-off, efficiency and resilience. And that's, well, that's one of the reasons that usually biologists use to justify so much redundance in biological systems. Have you checked whether the systems you're dealing with are, in spite of being efficient, are also resilient to small fluctuations in parameters?
Yeah, so thanks for the question. So as I mentioned, like in the beginning of the talk, so in the very beginning, when Barlow introduced this efficient coding thing, he said, like, redundancies were bad, right? So because then you don't have noise, so it's fine. But then when people started to look at noise in the system, then they said, okay, this is not really the case, right? We actually want to make sense of redundancies because in some cases, like we need that thing. So what we do now in the new efficient coding frameworks is to include noise. So this noise in the system itself, so in the neuron, and in the input of the signal that you have, help to understand what these neurons do under different noise conditions. So in principle, we don't consider the case in which neurons are perfect and they can just optimize the information. No, we always try to put meaningful noise to understand if neurons can do well. And also, in the direction of this question, going back to the connectome, we are trying to understand also, for instance, variability in the network and how we can understand this from an efficient coding perspective. So for instance, if you can create a specific neuron, if you can, to help both evolution and noise, if you can make this system, for instance, variable, right, to keep up for different stimuli ranges, to try to make sense of that. So that's something we are working now in the lab.
Hi. You said that you lose the advantage of having a diverse neural population when you use Gaussian random data instead of natural images. Have you tested whether the advantage is lost or kept if you use randomly generated images that preserve the spectral properties of natural images but are otherwise unstructured?
So far, we are only working with actually the power spectrum of the images. So we are not looking at the structure of the image itself. So we are not taking into account the phases so far. So the first thing we wanted to do was just to check if these correlations are enough to give us this result, right? And the next step is, of course, to test what happens if we send some specific noise function. So one of the reasons why we said we should go with this power law behavior is so that there was a paper by Mikko Jusola, and he was showing that if you have photoreceptors and then you look at the information rate of these neurons, when you send natural inputs and this random phase stimulus, they have a quite similar information rate. So we thought that this was a naturalistic enough system to test our hypothesis.
I have a question and a curiosity. The first one maybe is simple, but at some point you were showing in your slides a differential equation that was probably clarifying the model that you use, because I will have need that you explain it in order for me to understand, what you really do. I didn't get what type of model are you using in order to do all this beautiful work that you do. Can you go through the equation and explain us?
The one on the population model. So there are different ways of tackling this.
Where there were these variables H that you use that are the filters probably, right? There was a big equation that I wanted maybe that you commented.
This one, no.
No, before that.
Okay, so this is like the population, the color coding, this one.
Sorry. This one.
This one.
Yes. So can you explain us?
Yeah. So here, what we wanted to do was to investigate the activity of homogeneous populations. So we have this is like a voltage, the dynamics of voltage in these neurons. So we have a time scale of the excitatory populations, and this is one equation for one single population. So for instance, for red, so this is the voltage.
Yes.
This is like a Hodgkin-Huxley,
Yes.
exactly. So we have the external input. So this is going to be, for instance, the monochromatic color that we are sending into the network. And these are going to be the inputs from the excitatory and the inhibitory network.
Interactions.
So, interactions, exactly. So this is going to be from the horizontal to our neuron, and this is going to be from the different cone populations to this specific population.
And how do you decide? What parameters are those? What values do they have? How do you?
So what we do is to model these equations across the different parameters. So we play in the parameter space, and then we explore what are the more efficient configurations that we can have for this.
And what are the shape of the functions, Fi and Fa?
These are like sigmoidal functions. So we have a saturating output of our neurons. So yeah, there is no non-linearity here.
And then there is also another equation for the evolution of the horizontal cells.
See, this is like the horizontal cell equation. So here, the horizontal cells don't get input directly from the external environment. So this is not here. And these neurons get input from the cones, right? So they integrate information from all the cones, right? And there is a different parameter for each of the population, right? And then this is like the whole dynamical system. So if I have, for instance, a tetrachromatic system, so I'm going to have 4 of these equations, a total of 5 equations in the system.
Okay.
And then what we optimize is, in this case, is like are the weights of our networks to understand the encoding of this.
Okay, very good. So then I had another curiosity. Can I?
The curiosity.
It's just, I mean, I don't know why, but I mean, in most of these studies about the brain, usually they just look at one fly, one mouse, no? So do they have experiments in which they have social interactions like 2 flies, 2 mice together to try to see what happens when you put 2 of them together?
So I think, you mean, okay, so I think this is like the next question that maybe we will be able to understand and answer from with this connectome of the fly, right? Okay, no, I understand the question. So, okay, so for instance, for this, for these experiments in which they show like the responses of the neurons and like how they respond to the environment, they do it across many animals, right? But normally, so there are like difficulties at measuring like many animals because then you need to have like to really focus on the specific neuron that you want to measure. So your microscope is huge. Exactly. Yeah, I don't know if there is people doing that, but I think so. There is people trying to put in the like on the plane, so what the animal sees, other species. So for instance, trying to simulate what like the presence of other animals are. So for instance, in flies, you can do like behavioral setups in which you have like a fly, and then you put spots, and then that simulate flies, right? And other like more with flies, you can do many things. That's like and many times like not really nice things. So you can, for instance, like I don't know, put if you want to study like copulating behavior between flies. Sometimes what people do is like to put a dead female near to the fly, to the male fly to see what's going on. So it's possible, but I think at the collective level it's harder because then you need to keep track of the brain of many animals, and that's I think we hope. I mean I think that's the hope. Yeah.
Very interesting talk. I have 2 questions about noise. The first one, you told about a naturalistic perspective. So would be a naturalistic perspective of noise in your view? And the second one, and correct if I'm wrong, I understood that when you changed the noise distribution, you lost the differentiation from the multipolar or bipolar or unipolar cells. So at first, these 2 questions.
Yeah. So okay. So the idea is like if you have only Gaussian stimuli, right? So you don't have a lot of correlations in your input, right? So if, for instance, if you look at the power spectrum of Gaussian inputs, then you are going to find like a flat distribution, right? So if you go to naturalistic inputs, then you are going to see that correlations are important, and then you start having this power law behavior, right? So in many cases, you will see, so for instance, you can think about like pink noise and brown noise, right? So these are like variations of noise that happen in nature. And that is the thing that we want to incorporate here, right? So given that we have this type of noise in the system, then what happens? And then for many of these, like from pink to brown noise, we see this diversity, that the diversity of temporal filters improve the signal-to-noise ratio, right? But then when we put then the Gaussian noise, we lose this advantage. So the hypothesis is that if you don't have many correlations in time in your stimulus, then you don't really need like a fancy range of different temporal scales in your system to actually keep up with the information that's going on. So you maybe just need, I don't know, 1 or 2 neurons that are not really different in time, so in temporal properties to do that. So that's like the intuition behind the.
So you're considering the naturalistic perspective as the discrete noise, for example, the Poisson distribution. Okay. Sure. Thank you.
Thank you very much, Luisa. So if you change the environment, how long does it take for these neurons to adapt?
Okay, so this is, okay, so you are talking about adaptation. So these questions that the question that I'm talking in the last part of the talk is an evolutionary time scale, right? So I'm talking about, assume that you have fixed neurons with this type of temporal filters, and then you have this, right? So you are asking about adaptation, right? So we assume that neurons can also like do something like what's shown here, right? So we are not including this type of adaptation in the system. So we assume, so for instance, we are talking about like biphasic, right? Neurons that encode like for changes in contrast, and monophasic neurons that care about like actually sustained luminance information in the environment. And we expect that adaptation will not allow, like fast adaptation will not allow these neurons to be equal, right? So we are talking about like neurons that are actually far apart from the encoding perspective.
Okay, so my question is because of the following. So if you have a migrating species, it will spend some time in one environment and some more time in another environment, right? So it's expected that these neurons would have to be adapted to both situations simultaneously, or they would have to readapt every time that there is a migration. So is this type of adaptation, I know that this is not your field, but is this type of adaptation possible during the time scale of one life of the individual, or only evolutionary adaptation is possible?
Okay, so I think maybe one of the nice things about this, like a power law spectrum thing, is that you have, for instance, this scale-free idea, right? So if you go and change the environment, you still have these correlations in the system. So for instance, if your strategy is to have this diversity of temporal scales, like the system is going to be able to keep up with this noise in the system, like these different things. But if you only saw like Gaussian stimuli during these evolutionary times, I think it's going to be much harder, and then you need like these systems to actually be able to adapt and like develop these things. But I assume that if you already are in a naturalistic environment where you have these typical noise structures, then you are able to maybe, I don't know, I can't tell like how much time, but you can be able to evolve this.
Thank you.
More questions? I have a question related to that one. What these flies are old?
What?
Are they old? So these
Yes.
are maturated flies?
Yes.
What will happen during learning?
During learning?
Maturation? And what about aging?
Yeah, okay, so that's a tough question. So we will need a lot of machine learning things to understand how structure changes, and that's a really cool question we would really like to do. So we have some people looking at the connectome of larvae to try to understand this, how connectivity actually changes, right? I think learning is a much harder question because then you need to keep the connectome to understand like the structure like really in time, and that I think is a harder question. But what people are doing now is like to look at larva stages and try to like maybe make sense of this question.
And what you showed, depending on the noise and kind of structured responses, it reminds me a little bit about stochastic resonance and coherence resonance in nonlinear systems. Is there some work on that?
Oof,
You know what this is? Stochastic resonance?
yeah.
Where you have an input of a noise, a non-zero noise that maximizes an output. And you can also go for, if you do this in some kind of coupled systems, you can also generate some kind of coherence patterns without going into details.
But you mean, so what's the question that...
No, my question was, is there some kind of a relationship between these concepts that emerged in the 80s related to what you observed here in the fly?
Maybe, but I'm not
Okay.
completely
Okay.
sure about it.
Good. Time for lunch? Oh,
Thanks.
enjoy. Thank you.
Yeah.